Hello, everyone. My name is Wouter van der Bijl, and I'm a postdoc with Judith Menk at UBC in Vancouver. And welcome to today's EvoEco seminar. So before I introduce today's speaker, I want to briefly mention that we just released our new schedule, which is showing on the screen now. So for the rest of July and August, we'll be having talks every Wednesday. And we'll see what happens in September. We're not quite sure yet, to be honest. For this week, however, we still have talks every Monday, Wednesday, Friday, so our old schedule, which means that on Friday, we will have Owen Petchy talking, and he'll be speaking about classification in ecology and why ecologists should not divide things into groups. With that, I want to introduce today's speaker, Professor Chris Wheat. So Chris did his PhD at Stanford, working with Ward Watt. He then moved to the Max Planck Institute in Jena, in Germany, to work with Tom Mitchell-Olds. After that, he joined the Metapopulation Research Group of Ilkka Hansky, which is in Helsinki. And he's now an associate professor at the Department of Zoology at Stockholm University. So Chris's work usually involves the use of genomics to answer questions about ecology and evolution of butterflies. And these questions are about the wide range of topics. So for example, this includes the genetics of flight performance and dispersal, the coevolution between butterflies and their host plants, conservation genetics, as well as the evolution of color and life history. And today he's going to talk more about that last part, the evolution of life histories. So thank you, Chris, for agreeing to give a talk with us. And the stream is yours.
Wonderful. Thank you, Wouter, and thank you to all the organizers for selecting me to come on this venue to give a presentation. You've had a wonderful lineup of speakers, and you have a wonderful lineup of people coming in the future. So this is a wonderful contribution to the community, and I'm really thankful to be part of it. Today, I'm going to be talking about the evolutionary history of an ancient life history strategy. And as Wouter said, I study a diverse number of things, usually focused on butterflies. And before I get going, I want to do a quick introduction to my current lab and what we're doing. I have a PhD student, Hannah Dort, who's studying the chemical war between insects and plants. And she's looking at the gene family dynamics of detoxification of Brassicales plants. I have a postdoc, Rachel, who's just started, and she's quantifying alternative splicing in alternative phenotypes, trying to figure out how splicing is associated with stress and development. And Noah McKinnon, who's recently finished, was doing a lot of work on the immune system of butterflies, quantifying phagocytosis and PO responses, trying to figure out the evolutionary dynamics behind those. And the work I'll be talking to today is primarily the PhD project of Kelly Tunstrom, looking at the causes and consequences of alternative life history strategies. And this is just a picture of Vanessa Cardui using macrophotography, and I think it's quite wonderful. So there's that. So today I'm going to be focusing on life history and trade-offs. And I wanted to do a much more in-depth introduction to this topic because I find it fascinating. But I've cut things rather short, and I apologize for that. But in sum, life history evolution is focused upon the phenotypes across an organism's life cycle that affect the survival and reproductive success. And different types of life histories might be what species converge upon. There might be plastic ways to achieve different types of life histories. And these arise because of the limited resources that organisms are able to acquire during their lifetime. And they must invest in alternative phenotypes and essentially suffer those consequences throughout the rest of their life. And while these life history phenotypes that affect core aspects of Darwinian fitness are central to understanding the ecology and evolution of species, we actually know surprisingly little about their mechanistic basis and their evolutionary history. And one of the most inspirational books on that topic for me is the edited volume by Flatt and Hayland, Mechanisms of Life History Evolution. And if anybody's interested in these topics and a wonderful overview of the field, I really recommend that book for late night reading. It's quite wonderful and insightful. A lot of really good chapters from some great researchers in there. So in my lab, to deal with these type of questions, I'm focusing on a genetically determined alternative strategy that's polymorphic within a species. Not all alternative life history strategies are genetic, some are plastic, but here I'm focusing on one that's genetic and polymorphic in a species, because that allows me to actually start doing a lot of genomic dissection of the phenotype. Again, plastic and fixed traits can be more difficult to study, and that's why I'm focusing on the trait that I'm talking about today. And the trait that I'm talking about today is exemplified with these two species. In the top row, we have representative butterflies of Coleus eurythemi. This is a butterfly that's common in North America. And in the first column, we have an Alba female. She's white. And we also have a colored female. She's orangish yellow. And you can tell they're females because in the border of their wings, they have spots. And on the far right, you have a male. All males in these species are a fixed color like this. And these polymorphic white females might be anywhere between 5 and maybe 20% out in wild populations. And the second row is looking at a very similar species in Europe, Coleus crocea. The genus Coleus has about 80 species. And this white phenotype, this Alba phenotype, it's only found in females. It's polymorphic in about a third of these 80 species. And some classic genetic work has dissected it out to being a Mendelian trait, so one locus of very large effect, that's autosomally inherited, so it's not on the sex chromosome. And just to really reinforce the architecture of this trait, this is a genandromorph of Coleus eurythemi. So the left half of this bug is a male, and the right half is a female. Genetically, this bug is carrying the Alba allele, but the male side doesn't show the phenotype. But the right side, the female side, does show the phenotype of Alba. And that just helps you perhaps get a firm understanding of what this trait looks like when it's segregating. The reason we focus on this trait, although it's a color phenotype and it's quite beautiful, Alba is actually a female-limited alternative life history strategy. What do I mean by that? Well, these two color morphs differ in some really important aspects. The Alba phenotype, they have a faster development time. They emerge as adults with a larger fat body. They have a higher fecundity, but they have a lower attractiveness to males. So effectively, males are more willing to chase and mate with orange females. So this color difference is actually a trade-off between attractiveness and fecundity. So here's some data providing an empirical basis for what I just said. This is for Coleus eurythemi, again, the North American species that I'm talking about. And here we're looking at the number of eggs females are laying after mating, egg numbers on the y-axis. And what you're seeing are the average of around 10 females and the number of eggs that they're laying throughout their lifetime. And these are for orange females. And now I'm showing Alba females. And you can see that between 6 and 12 days, Alba females are having a much higher fecundity. Also in North America, where a lot of this research has been done, focusing on the Alba phenotype, back from my old advisor, Ward Watt, they also looked at male choice in terms of which females they preferred in the wild. And they went out to the wild, out in the Central Valley of California, and they attached small strings to Alba and orange females, kind of let them fly around, and watched which males would come and which ones they would actually prefer to interact with. And the table that I'm showing on the right is highlighting how during many different days and in the morning and the afternoons, there were a lot more approaches to Alba females, or rather orange females, compared to Alba, right? So males are definitely a lot more interested in orange females. We've been trying to do similar work in Coleus crocea, effectively the European equivalent of the North American species I was just talking about. And we were just in Spain last year. I wish we were going this year. And we took actual butterflies and put them on little stands out in the field and put a camera up. And I'm just going to show you this video. We would set the camera and let it record. And we did this in multiple places and multiple fields. And you're going to see a male come in and he's going to inspect each one of the models. And then he flies away. And so we ran this for a couple days in a number of different settings. And our preliminary results show that out of 40 trials, males only approached Alba in 2 cases, right? So even in this European species, it's clear that males prefer orange females. We've also done a split brood design where we've taken in some females, we've gotten their eggs and we've reared them at different temperatures where they're segregating, either having Alba or orange offspring. And we measured the total fat content using lipidomics. And what we can see here is that offspring that were reared under cold conditions, the females emerge as adults, 2-day-old adults. They have much higher, larger fat bodies compared to orange females. And this is consistent with what was found in the North American taxa. Just to kind of emphasize, this life history trait appears to be similar in different taxa that, as you'll see, have been separated by a long period of time. So Coleus eurythmi, Coleus crocea, they look similar. Their Alba phenotypes appear to be similar. How did this alternative life history strategy evolve? And these are some of the questions that I'm going to touch upon today. I'm going to talk about how we've identified the locus of Alba. I'm then going to place that into a larger evolutionary context of the genus. And then we're going to talk about finding the genetic basis of Alba in some other species. And then trying to look at how that came to be, whether this was independent evolutionary origins of Alba, whether this is due to introgression, whether it's due to long-term maintenance. Our ultimate goal is to get at the questions on the right side. How long can these type of alternative life history strategies be maintained within a species? What determines the frequency of these alternative forms? Why are some populations only 5% Alba? Why are some 20? Why aren't some 80? Some species within this genus are entirely Alba. They've gone to fixation. Why? Sometimes it looks like it's been lost. Why again? So by trying to get to the genetic basis of this trait, I want to get at these long-term questions to make that genotype, phenotype, eco-evolutionary connection, because I think that tells us a lot about the evolutionary process for a phenotype that's really important. If you go to a museum and you get into their collections, and you open a drawer of Coleus, you'll usually see something like this. It'll have a bunch of males and sometimes a bunch of Alba females. And this is just a population sample of Coleus valteri. And we're going to go in close to these butterfly wings. In fact, we're going to get right up on the scale. And what we're going to do now, you can see the ridges in these scales. And we're going to zoom in on a couple of those ridges using a scanning electron microscope to see what's giving rise to these colors. And what we see here is that between these ridges, we have these cells. And in those cells, there are these rice-like granules. And those are actually pigment granules in these wing scales. And that's what's giving rise to these colors. Pteridine pigments come from pteridine butterflies or hyarid butterflies. And there's a number of different pigments that give rise to the different color variation of orange, yellow, red, and colorless. So most of you are probably very familiar with the little white butterflies you see out in people's gardens eating their cabbage. These cabbage butterflies, if you were to go in and look at their wing scales, they're chock full of these pigment granules, but they're full of leucopterin, which is what's giving rise to their white color. When we go in and look at the Alba or the orange phenotypes, we see something very different. While the orange genotype here on the right has a lot of pigment granules, it essentially has sepiopterin giving rise to the orange, the Alba phenotype doesn't have leucopterin. It actually lacks pigment granules. So it's a very different way to achieve the white color. If we chemically remove these pigment granules from an orange wing, we can actually recover the white coloration, showing the association between having these pigments and coloration. So I'm going to start off by talking about how within Coleus crocea in Europe, we identified the basis of Alba. This was the PhD project of Alyssa Woronik when she was in the lab. And we essentially did a lot of work on this species for about 5 years. And that recently got published. We generated a genome for the species. We did QTL mapping for the trait. And we also did a genome-wide association study, sequencing 15 orange and 15 Alba females of diverse family backgrounds. And the sum of all that was finding this one peak. And we know that we should find one region in the genome, again, because it's a single locus Mendelian trait. Turns out that Alba is appearing because of what looks like a series of transposable element repeat events that have inserted within this region of the genome that's unique to Alba. And that's occurring next to a gene called BARH1. And BARH1 got us really excited. It's required in the developing eyes of flies, and it's been well characterized there from an evo-devo perspective. But when you knock it out in flies, their eyes lose their red coloration. They lose, actually, their pigment granules, and you get effectively these type of white eyes. And that seems somewhat similar to our phenotype, so we were very excited. BARH1 is a homeobox transcription factor, and it's actually a repressor of other developmental genes. We thought that was pretty interesting. So with the help of Mike Perry, we developed CRISPR-Cas in our lab, and we knocked out the BARH1 gene. This is a Coleus crocea healthy individual, wild type, with their beautiful eyes and pink coloration. They're a cute little bug. When we go in and do CRISPR, they're not so cute. What happens is that we're getting a mosaic of bar knockout across the eye. And what we think is happening is that that's preventing normal bar functioning. And that's essentially causing regions where pigments are not forming, kind of equivalent to the Drosophila eye. And this is leaving regions that are black. And so what we have here is a bar mosaic across the eye. And we think the absence of pigmentation. When we actually, after injecting well over 1,000 eggs, we got this working in an Alba female background. And this is what we found. So the wing is Alba, but you have this mosaic of the recovery of color, the recovery of pigments, the recovery of pigment cells or pigment granules, right? So we think that bar is a repressor actively working in this Alba background. And because we're now knocking out bar, it can't repress anymore. So we get the recovery of pigments. And if we zoom into these wings, in fact, this is what we see, that the absence of pigment is due to the absent, the white scales are due to the absence of pigment granules in this mosaic. Okay, so we've identified the locus of Alba in Coleus crocea, and we do want to look at it in Coleus eurythemi and eventually across the whole genus. So we need to start developing an understanding of the evolutionary context in which Coleus crocea falls. And to do that, we've sequenced about 22 individual genomes from close to 20, maybe 21 different species at this point. First thing we did was extract out the mitochondrial genome from all the species we sequenced. And we took out all the coding regions of genes and aligned those together. That's about 10,000 base pairs of coding DNA. And we ran a maximum likelihood analysis on that. And we used as an outgroup a calibration point with Zerini Sassonia to try and put some type of time estimate on the age of this genus. Now, the tree itself has some problems, but the major features are actually very important. First, we have a root of Coleus that's about 4 million years old. And that's breaking off between South American taxa and then the European and North American taxa up here. And the root of what looks like the European and North American taxa looks to be about 1.5 million years old. Because these butterflies have 1 or more generations per year, this is actually 1.5 million generations ago, if not more. So that's our current estimate, at least from the mitochondria. We've also gone in and extracted out the exons from all the genes in the genome and took one exon per gene. On average, they're about 150 base pairs in size. And I only chose exons that had variable sites. And now I had exons essentially in a context of the genome, and I could generate the following. So I generated a set of gene trees and took the consensus of that, the species tree using Astral for the sex chromosome, which I'm showing here on the left. And I also did that for each of the 30 chromosomes in the genome. There's about, I'm getting about 320 exons for the Z chromosome and about same number for all the different autosomes. And what we can immediately see is that there's a lot of discordance across all these different exon trees, if you will. But some really important features come out of this. So if we focus in on the autosomal exon tree here, this is actually grouping things pretty well in the regional clades that we a priori would expect to see. That the deepest branch that we have within this clade is for South America, colored in purple. Then we have a European clade, which is pretty coherent. We then have an orange, what we're calling a whole Arctic clade. And these are species that are occurring at high latitudes that are circumpolar. And then we have at the bottom here, the North American clade in blue. And I'm leaving in all of the alternative chromosome level trees just to show the discordance across this. When I run a gene concordance analysis, that's very easy to do on IQ-Tree, and it's a wonderful tool. It allows you to see past bootstraps and posterior estimates, how much each exon is supporting every node in this. And the support's actually pretty low. So what this Astral kind of species tree, I think, is telling us is that it's the best you can get with the data. But it suggests that there's a very strong impact of short branches because these guys appear to have radiated pretty quick. There's a lot of incomplete lineage sorting and potentially introgression taking place here. And keep in mind that even though I'm only showing about 20 species here, it looks like within the last 1.5 million generations, about 70 species have been generated, right? So there's been a lot of speciation going on in here. Some very short branches, a lot of incomplete lineage sorting. And many of these species are known to hybridize. So introgression is, of course, expected as well. So here I've cleaned up that tree a little bit, and I've put a little time estimate here. That gives us an estimate of when Europe and North America likely last shared a common ancestor. And I've showed you that we know the genetic basis of this Alba phenotype in Crocia. But now I want to start thinking about, well, what's happening in North America? What's happening in Coleus eurythemi that's been long studied for the Alba phenotype? Are these independent evolutionary events that give rise to Alba? Or is this an ancient origin that somehow has been maintained within this genus? Well, we're now going to move on to assessing the locus across some other species. And now we're going to focus on finding the Alba locus in Coleus eurythemi. We sequenced the genome of Coleus eurythemi using PacBio sequencing. We generated about 140x genomic coverage, and that wonderfully assembled into 123 scaffolds. I've never seen an assembly work that well before. It was lovely. Using a linkage map, we were able to assign those scaffolds into 31 chromosomes. We then polished it a bit, and we got some very good BOOSTGO results that said that we had a good haploid assembly with very good gene content. Here I've just done an alignment between Coleus eurythemi and the sister genus Zerini sasonia. And all these lines from the chromosomes of eurythemi over to the scaffolds in Zerini, they're very solid lines. The overall genomic structure is very similar with Zerini, and Zerini is very similar to Heliconius. Coleus genome is nothing exciting. It's a very standard butterfly genome, which to some extent is a good thing. We then used a cross between Coleus eurythemi and a sister species to it, Coleus phallotici, that was segregating the Alba phenotype. And with the F2 offspring, we could then track what chromosome was associated with the Alba locus. And using this approach, you can see in the bottom panel here, that we have this one chromosome 3, that has a very high LOD score associated with the Alba phenotype. And that's great. That tells us that we know what chromosome to look on. But in order to get inside that chromosome, we needed to do a genome-wide association study. Unfortunately, this cross was based on a female informative cross where you don't have recombination, so we can only get chromosome level insights. The top panel is showing the results of this GWAS. We did 15 orange and 15 Alba females again. You can see this nice peak here on linkage group 3. And now we're zooming into that peak, and then we're in the middle of that peak region. And what we have mapped here are reads mapped to the Coleus eurythmy genome from an individual that's an Alba and an orange. And we also mapped Coleus crocea Alba and orange individual to the same genome. And at this part of the map here, this is the bar H1 gene region. And you can see that these little peaks here are the read depth. So this is the depth of all the reads that are mapping in this region. And you can see that both species and both morphs, everything's mapping to that region pretty well, and they should. The bar H1 gene is fairly well conserved. Nothing exciting is happening in that gene at all. But as we move 3' away from that gene, this green region is highlighting a unique genomic content to the Alba phenotype within the Coleus eurythmy genome. What you can see is that there's Alba reads mapping to that region that the orange individual does not map against. So this suggests unique Alba content here. But when we compare that to what's happening with data from Coleus crocea, the read depths here get crazy, right? What's happened here is that this is actually a repeat element that's only found in Alba in Coleus eurythmy, but that repeat element is actually diversified in Coleus crocea, and all those different copies, when they're being mapped to Coleus eurythmy, have no other place to go, so they stack up here. So this is a repeat region that we've colored in gray. In pink, however, is something very exciting. This is a region that has unique content that's only found in Alba individuals, not in orange. And that's also similar when you look in Coleus crocea. And so this is our first hint of about a 1 kb region that looks like it might be involved in the Alba phenotype itself. And we're going to focus on this pink region in a couple of different analyses. But what this tells us is that it looks like very much that the genetic basis of Alba in North America, Coleus eurythmy, is the same basis of Alba in Europe, Coleus crocea. So Alba is the same in these two taxa from Europe and North America. But what about these other Alba species? Remember, we generated genomes for about 20 different species. So our question now shifts to, well, is this Alba phenotype, is it being maintained due to negative frequency dependent selection in these different taxa, or is it moving around perhaps due to introgression? Among these taxa that I have here in this phylogeny, I'm going to quickly put up a little cartoon that shows you how we've sampled across the Alba and the colored phenotypes. This is not what these butterflies look like, but they just tell you how we've been sampling Alba and orange phenotypes from across this diversity of Coleus species. So within Europe, we have a number of Alba and orange, also in North America, also in the whole Arctic, okay? So we're sampling both these phenotypes throughout the range of the species. Although in South America, we only have orange individuals, although it is known that Alba occurs down there. So our question then becomes, well, is this Alba insertion that we think is actually associated with Alba, but we're only seeing that in two taxa? What if we look across all these other species using the genomic data that we have? Is the Alba insertion only found in all the white morphs? And so Kalle did a really interesting analysis here. Because he identified that 1 kb region that looks like it's associated with unique content for the Alba allele that was shared in Europe and North American eurythmy and crocea. he tried to see if that is actually true across all these species that we have data for. And so what you see here is a phylogeny. And this TE insertion region, this is that pink region that I showed you before that was the unique Alba content. But that's a hypothesis for eurythmy and crocea. And here we're looking across all the genomic data mapped to the Coleus eurythmy genome. And we see that all these species that are actually orange individuals, they're actually not having any mapped content in this region. These histograms represent the number of reads that are mapping in there. But every Alba individual, they're actually having reads map into that Alba insertion region. In fact, every white morph, which we actually think are Albas, all have content mapping to that window. In order to compare this window that we're looking at here across the entire genome, well, we repeated the same type of analysis for about 550,000 windows across the genome of the same size. The way we did that was as follows. On the right side, if you can see my pointer, you can see that we could score each of these results of the mapping in a binary fashion. These would all be zeros. This is a 1, 0, 1. And it's not a 0 and 1 based on color. It's based on reads mapping within the window, right? But this is how, for this one window, we would score it. And in this case, every white morph actually has a 1, and every orange morph actually has a 0. And if we take then the mean of those totals and look at the difference, we can actually plot that. And so what you see is that the vast majority of windows in the genome are identical between the two groups. It doesn't matter at all. But it's only when we actually look out at this tail here that we find complete concordance with Alba. And that's the window that I'm showing you here, right? And so what that's telling us is that the Alba insertion is unique in the genome, and it's unique across all these species that have Alba. So only white species have reads that are mapping to this region, and the colored individuals never do. So this suggests that Alba certainly does have a unique basis and that we have a pretty good handle on it. But how did Alba get into all these different species? Well, we need to look at introgression. And to do that, we started working with D-statistics. And the analysis that we used was an F4 branch analysis. This essentially assigns the F4 admixture results to nodes within a tree. And we're using the exon consensus tree that I showed you before. And this is perhaps not the most intuitive figure, but what it's showing us is that cells in here that are colored dark have a lot of introgression that's associated with the base of a node here. And this is the base of a node that gives rise to a clade of butterflies that are fixed for Alba and that are circumpolar. And those look like they have introgressed to a large extent with these species up here, and those are North American taxa. So this is a clear suggestion that there is a species with fixed Alba that has introgressed into a regional clade, and that could be the origins of how Alba is actually getting into North America. Yeah. There's also some evidence of this taking place within European taxa. When we tried to take this type of approach, and instead of do it at the genome-wide level that I'm showing here, but go in and actually look along chromosomes and across the Alba scaffold itself, we couldn't detect any of these type of results. In fact, it was a little frustrating. We think there might be really good regions for that, that the Alba locus itself is very narrow. It's essentially a copy number variant. You either have it or you don't. And the shoulders of that might be very, very narrow. And other signs of introgression quickly get recombined out of the population. But that's a working hypothesis. We tried to get at this issue in another way. What we did was we extracted out the Alba locus, that roughly 1 kb region that was specific for Alba content. And we did that for all the species that are white, or essentially Alba, that have this. And here I'm showing a phylogeny of the Alba region. And what's nice about this is that it's consistent with what I was showing you in the previous analysis. Again, here, ancestor to the nasties clade is introgressing into the North American clade. And what you can see here is that all of the Alba alleles within Coleus eurythemi, they're most closely related to this nasties clade, right? So that's consistent. It looks like some ancestor of nasties or nasties itself somehow got its alleles for Alba into Coleus eurythemi. It looks like they've been in Coleus eurythemi for some time because we see some structure across the alleles that are segregating for Alba within this population. That can happen if they've been in there segregating, mutating, essentially having their own evolutionary history within the species. That's also happening within Coleus crocea. Again, this genomic data is from our GWAS analysis, so we have lots of individuals we've sequenced for Alba. So it looks like within both those species, Alba's been around for a while and it's been able to mutate, and those have been maintained. What's also interesting is that in North America, it looks like Polydny might have donated Alba into Philodicea. Philodicea is very closely related to eurythemi. So two closely related taxa, looks like Philodicea didn't get Alba from eurythemi. It looks like it got it from a completely different species. It looks like Coleus polano, another circumpolar species, donated Alba into some European species that are fixed for Alba. So we're definitely seeing some results of introgression, donation of Alba alleles. Looks like these might be old. Not exactly sure when it happened at this point, but it's very encouraging results. So in summary then, this alternative life history strategy called Alba appears to have evolved only once. And it looks like that was at least 1.5 million generations ago. Because the South American clade of Coleus, we know also has Alba, it's perhaps parsimonious to think that it's the same basis, but we haven't shown that. But if it is, that would mean that Alba might have been around for about 4 million generations. If you think about the generation time of humans, somewhere between 20 and 30 years, right? We're talking a very long time, right? I mean, we're talking if it's 20 in terms of human context, right? Would that be 80 million generations ago, right? Or 80 million years ago, if you put this in a human year context? I think it's important to put these time estimates in a generational context so that you can think across taxa. And when you do that, it appears that Alba is actually quite old. It looks like it's moving across species via introgression, right? And that the donor species of Alba within a continent might be diverse. We find evidence of that. We also find evidence that Alba, once it gets into a species, it's maintained for a long period of time, long enough for these alternative Alba alleles to diverge from one another. Those could be independent introgression events, but we don't see a lot of introgression, say in Coleus crocea in Europe. Maybe we haven't sequenced all the taxa that we need to pick up on signature, but certainly the alleles within the species appear to be diverging from each other. And you would need time for that. It also suggests that Alba might have introgressed into some species and gone to fixation. Right? So this set of findings that I've showed you today help us move forward to get to the questions that we really want to dig into, because I think they're important questions. How long has this Alba polymorphism been around? Has it always been maintained as a polymorphism? How long can it be maintained as a polymorphism within a species or within a lineage across several speciation events itself? A lot of the other examples where we know large kind of alternative life history strategies like this, they don't appear to be very old. So I think this is a fascinating finding. Why is it old in this clade? And what determines the outcome of Alba once it gets into a species? Why in some species does it look like it's been polymorphic for a long time? Why in other species might it be going to fixation? Or why might it be lost and perhaps resistant to the introgression of Alba alleles? These are really good questions. It allows us to start connecting genotype to phenotype to ecology and evolution and the dynamics of those. which is where I really want my research to spend some time. Finally, although we've identified the locus that's associated with the Alba phenotype, and bar H1 looks like it plays a large role in the phenotype, how does that Alba locus, perhaps even that 1,000 base pair region that's unique to Alba across all these species, how does that give rise to the pleiotropic effects across these different species? Right? Is it life history trade-off? Is the alternative life history strategy, is it the same in every Alba species? Is it evolving in a different way? What other genes might bar H1 or other transcription factors in this region be affecting? We don't know, but that's exactly what we're trying to work on now. I did not do this work alone. This was the result of Team Alba. It's a diverse, great group of colleagues. Kelly Tunstrom has done a lot of the D-statistics and analyses that I showed here. Joseph Hanley did a lot of the QTL analyses. Pasi Rastas helped us get our chromonome. Anton, Andy, and Akito been a huge help in Sean in securing samples of diverse coleus species. Vincent helped out with a lot of the molecular work, as did Ward and Adam in providing contextual frameworks for understanding this, as well as critical material for developing our genomes and crosses. And Arnaud has been a great, essentially co-leader on this project on his side of the Atlantic. And with that, thank you very much.
Thanks, Chris. That was super interesting. I'm sure we'll get a bunch of questions. I'll start with one of my own questions. So since it's an autosomal locus, do you have any idea how it's expressed only in females?
Our hypothesis is that there might be a transcription factor associated with this Alba insert that could plug into a sex-specific transcription factor like double sex. And we have some preliminary evidence that suggests that, but we're a little hesitant to jump up and down about that now.
Fair enough. Something to look forward to then.
Yeah.
So some questions from the audience. Have you considered running specific tests for negative frequency dependent selection within the genus on the Alba region? And also looking for any signals of trans-species polymorphisms?
Yeah. So we're definitely interested in trying to use the population genomic data we have to see how unique Alba may or may not be in terms of the age of alleles that can be maintained within a species. That's a great question. And I've essentially showed you chapter 1 of Kali's thesis. That's kind of chapter 2. In terms of trying to estimate negative frequency dependent selection for these species, that's exactly what we'd love to do. I should be going to Zurich this fall to work with Hana Coco to develop those models to try and figure out what we do and don't know and how hard it might be to maintain this. We have a diversity of field collected data across years and sites that give us estimates of frequency. One of our sites, it looks like not only is Alba at a constant frequency of around 5%, it looks like it's slightly increasing. We've been trying to estimate the frequency by genotyping males. So that way we have an unbiased estimate of Alba frequency in the wild. Not that way we have a handle on the locus. That's working really, really well. But some of the traditional methods that have been used to detect negative frequency dependent selection, even just working at the phenotype, like in Ischnura elegans and some other phenotypes, they kind of have a boundary dynamic where you need to look across enough populations and enough years where the frequency is kind of bouncing back and forth, and that gives you the power to detect something maintaining that. We don't have good enough data for that, and because our trend is slightly increasing, it's not bouncing around in a way that would be amenable to those models. We've tried, but currently that doesn't work.
Thank you, makes sense. It seems maladaptive for males to discriminate against Alba females, given that they are more fecund. So does male discrimination have to do with avoiding hybridization? Are Alba females less distinguishable across species? Or are there more obvious costs to males for mating with Alba females?
I think males are trying to mate with whomever they can. When people have gone in and looked in the abdomens of females, you can count the number of times they've mated. This is called a spermatophore, and it's something that's kind of unique to butterflies. These butterflies will probably mate upwards of 2 to 5 times during their lifetime. And when you go in and count the number of spermatophores that orange and alba females have, there's not a significant difference, right? So it's not that alba females are getting less mating, although that was one of the things that we were thinking. The literature doesn't support that. Rather, this is also something that's probably similar to what's going on in another female limited polymorphism system, Ischnera elegans, where females get harassed a lot by these males, to the extent that they lose time realizing their fecundity. Coleus females, when they're chased by males and they don't want to mate, they do an escape flight, which goes up into the sky until you can't see them anymore. It lasts for minutes, maybe 5, and then eventually the male comes down, looking disheartened, I guess, and the female comes down some other part in the field and goes about her business of nectaring and laying eggs. So alba females could directly benefit from being less attractive because they're not getting harassed as much. So they have more time to lay eggs and forage without having to avoid males. And that could be a huge part of their energy budget. Ward, years ago, did some modeling for this, and it looks like, given the densities and assumed interaction dynamics, this could definitely be a powerful driver in this system. But it's more work that needs more modeling and more empirical data from the field. And that's exactly what I'm going to be trying to work on with Hannah this fall.
Exciting. So to stay with ecology for a bit, do you have any more information about predation? So someone asked, I would imagine that the alba form would be more predated since orange looks aposematic. Would that influence the fixation of the alba allele in some species?
Great question. Absolutely great question. I don't have that data, and I don't recall seeing any data like that on predation. People had hypothesized in the past that perhaps this white form was a mimic of some of the white cabbage butterflies, under the assumption that the white cabbage butterflies themselves were perhaps aposematic and contained some of the toxins that they get from their host plants. Those are some old theories. We know today that the cabbage butterflies do not sequester, and it doesn't look like they're an aposematic color, at least in terms of that type of chemical dynamic. And it doesn't look like there's differences in predation. I think Ward actually did some old studies on that as well. So I don't think predation is what's driving this. I think the earlier work that I showed that identified cold as being a dynamic in terms of fat body differences, that in warm habitat with high densities, alba might be favored to some frequency, and that might depend a lot upon the density of the population, which might change during the year. But in colder habitats, alba might always be favored. And it is true that most of the species that have fixed alba phenotypes are associated with higher latitude and colder habitats. And so you might have some interaction between interspecific dynamics as well as ecological dynamics that play out into which species have maintained polymorphisms versus alba fixations. And we're excited to track that down.
Yeah, so maybe you can say a bit more about why you think that the fitness of alba is linked to temperature across Europe, for example.
Well, yeah. Both in eurythmy and in Crocea, the fat body is larger when they're essentially developing as pupae under colder temperatures. And that might provide them more resources in a habitat that might not have as much flight time or as much resources during the adult stage of their life. And therefore, you might expect that also to correlate with colder habitats. If you're a mountain butterfly, if you go up to the Alps, there's Coleus facomini. They live close to 2,000 meters, and it's windy, cold, highly variable habitats. And so, if you have to spend a large fraction of your day gathering resources versus just laying eggs, having a larger fat body reserve might allow you to exploit good conditions better than if you had to forage. And that could kind of maybe push things over the edge and drive alba to go fixation in those habitats.
Yeah, makes sense to me. So a bit more genetics then.
Okay.
Someone asks, as it manages to jump to other species, do you consider the 1 kb Alba region to be a selfish genetic element or parasitic DNA?
I haven't thought of it that way before. Do I? What's the utility in thinking of it that way? I don't know. I'm gonna have to chew on that. I like it. I've never had a selfish genetic element in my lab that I know of. So it'd be fun to have one. I don't know.
All right, fair enough. So do you think this introgression dynamic, is that important to the maintenance of alba in species? Like, do you think it like periodically gets lost and then you need introgression to reintroduce it?
Yeah. So people have hypothesized that in other systems, there was a nice review paper that just recently came out asking the question about what maintains polymorphisms across species, looking at these kind of large effect polymorphisms and how they stick around, how they make it through speciation events and persist within a clade in ways that don't fit a simply neutral model. And it definitely could, it absolutely could be. The one thing that the preliminary data that we have, when we're looking at least coleus eurythmy and crocea, because the alba alleles there appear to be showing some divergence within them, that suggests that they've been there for a while, right? So that looks like a somewhat stable system. There could be other systems where alba is very rare, and maybe it comes in and goes out. And so although about a third of the species have alba polymorphisms, or actually perhaps more than that, sometimes that's really, really rare. And it might be that it's rare because it gets lost, because it never makes a high enough frequency to be retained through chance events. Maybe it doesn't have an advantage, and those represent hybrid individuals that were hybridizing with something carrying the alba. So I think that introgression could be a powerful way of bringing this back into taxa, but we haven't found a species yet where it looks like that's playing out. There certainly could be one, and yeah, we'll keep our eyes open for that.
Yeah, I guess you'll figure out in the coming years whether it goes locally extinct and things like that. Yep. A question about the visual system of butterflies then. Could the lack of attractiveness of alba females be a constraint of the visual system? Do we know how coleus eyes respond to the different phenotypes? And how do things look in the UV?
Yeah. UV aspects of butterfly wings is fascinating and understudied. The excitation response of males, to what extent that might differ with the two different females. Does bar play a role in males at all in terms of what they perceive? Because we know it is associated with eye development. Or the alba insertion? We don't know. I think that's an excellent question. I know colleagues have been thinking about it. We have a new professor in the department, Emily Baird, who has the tools to get at that. I know that Nate Morehouse has investigated some of those issues, but I think that's still very much an open question.
Yeah, it seems there's a lot of evolution going on in opsins in butterflies anyway.
Absolutely, they're incredibly diverse.
It might be quite interesting whether species that have lost one of the mores actually, whether you see changes in the visual system as well.
Absolutely, and also just thinking about choice, right? So in species where they're predominantly orange and you have a low frequency of alba, males clearly prefer orange. Fine. Well, if we go into an alba fixed species, do males prefer alba? Or do they still wish that there was orange, but they just don't see them because of the potential spatial temporal offset of where that species occurs? And that's something we would love to do if we could get up to the mountains and get Facomity back in the lab. But this is not the year for that.
Yeah, sure thing. So you mentioned some other species that have white polymorphisms. Are they always in butterfly and moth species, or are they also found in other insect taxa?
Yeah, so I'll get at that question gradually. So Nate Morehouse and a student of his, Emily Lamelli, I think, what's her name? They did a beautiful study trying to look at Alba as a polymorphism across the family Pieridae, trying to look at the white species and how diverse they were. And one of the things that they found is that a lot of species across Pieridae appear to have this type of Alba polymorphism. Whether it's orthologous to what's happening in Coleus, don't know. But it looks like, so here across this phylogeny of the different genera within Pieridae, in green are the ancestrally reconstructed estimates on nodes for which taxa would have had Alba as a polymorphic state. And it looks like this type of polymorphism is widespread. To what extent this is always due to changes in pteridine content of wings and changes in the granule content, and something having to do related to what we're finding with bar H1 and Coleus, that doesn't appear to be the case. We've done a lot of scanning electron microscope work, so that doesn't appear to be true. But thinking in this larger context, it made me kind of wonder a lot about color and life history trade-offs. I think the vast majority of our field, when they think about color, they think a lot about camouflage and they think a lot about mimicry and those type of dynamics. But I'm starting to think a lot now about cost and investing in color and what that might mean for other aspects of life history and trade-offs. So that's within Pieridae. Certainly across butterflies and moths, there are lots of white morphs. Those are not due to pterins. Those are usually due to changes in omochromes. You usually only find these pteridine pigments within Pieridae. But I was recently over at the museum looking at the microsculptures exhibit, where they have all these crazy macro photography detailed images of many different types of beetles and whatnot, and many of them are white. They have scale-like structures, but they have a very different, I think, evolutionary basis, and it's just not orthologous at all to the way that I'm used to thinking. But certainly there's white morphs across many diverse insects, absolutely.
Yeah, so to follow up on that, you mentioned that some of the white butterflies, like the ones you study, just have a lack of pigment granules, but some other species have white pigmentation to make the white color, right?
Yep.
So are these whites actually the same, or are there differences in the white color? So for example, is one of them more UV reflectant than the other?
Absolutely. And we haven't quantified that. I'm pretty sure that has been done. And I think that the lack of the pigment granules might give rise to potentially more of a UV reflectance, but I don't recall that literature as well as I should right now. But all white is not equivalent. Absolutely. If you tried to do a detailed spectral analysis of this, I'm sure they would look different.
Great, thank you. So I think I've grilled you enough for now. So we'll keep it to this. There's a few more questions on Slack that you could have a look at. I think Colin will also be in the chat. So if anyone has more questions, feel free to ask them. And I'm sure it'll get answered at some point.
Yeah.
Thanks again, Chris.
Okay.
I really appreciate it.
Yep.
So for everyone else, let me just show the schedule again. So on Friday, same time, same place, we have Owen Petchy talking. And then I hope to see you next week as well, when from that point on, we'll be only showing seminars on Wednesdays. Yeah, stay safe and goodbye.